Imagine a microscopic spy thriller happening right now, just, right beneath the surface of your skin.
Oh, yeah. Like a full-on infiltration.
Exactly. Like we're talking about an enemy agent infiltrating a highly secure facility, deploying a chemical jammer that completely blinds the security cameras, and then
Just walking right past the heavily armed guards.
yeah, just strolling right by.
And I think the most terrifying part is the guards don't even know they've been blinded.
Right.
Like they are physically standing there, completely oblivious to the threat that is just destroying the building around them.
It's wild. Usually when we talk about a medical diagnosis, there's this expectation of like mechanical precision.
Sure, yeah, like an X-ray.
Right. You break your arm, the X-ray shows that jagged white line, and the doctor just points and says, there it is, broken.
That's simple. Easy to see.
But then you step into the world of cancer immunology, and suddenly that X-ray machine is useless. We are looking at an invisible battleground where our own troops are actively being manipulated.
Completely manipulated.
So welcome to the deep dive. Today, our mission is to uncover a specific, pretty devastating medical mystery. Why does breast cancer become so incredibly difficult to treat when it spreads to the skin?
It is a critical question, honestly. When that skin invasion happens, the survival statistics just drop drastically.
Yeah.
And for a long time, the underlying mechanism of that failure was just, well, a black box.
But we are going to open that black box today. We are dissecting a really remarkable 2026 study published in the journal Cell, Death, and Disease.
A really great paper.
Yeah, it's by a team of researchers in China, and the title is Prostaglandin E2 Induces Dendritic Cell Dysfunction in Skin Involvement of Breast Cancer.
It's a mouthful, but we'll break it down.
We definitely will. Our goal is to figure out exactly how these tumors deploy those, chemical jammers we just talked about.
Right.
And even better, we'll explore how the mechanism behind a common, everyday arthritis painkiller might hold the key to turning the security alarms back on.
Which is just wild to think about.
It really is.
Yeah.
So to help decode the incredibly dense immunology and metabolomics in this paper, I'm thrilled to have our expert guide here.
Glad to be here. The data in this paper is stunning because it spans the entire biological spectrum.
Like from start to finish.
Exactly. We're going to trace this phenomenon from live human tissue samples down to the molecular soup tumors secrete, over to mouse models, and then finally out to massive global bioinformatic databases.
Okay, let's unpack this. Let's jump right into the clinical reality first. What exactly are we talking about when we say skin involvement in breast cancer? Because I think, most people imagine breast cancer as a localized internal mass.
Yeah, that's a really common misconception. Skin involvement, or SI, occurs when the breast cancer tumor extends outward and physically infiltrates the overlying skin.
Oh, wow. So it literally breaks through.
Yeah, exactly. Clinically, a doctor might see these rigid skin nodules, or a condition called carcinoma erysipelatooides.
Carcinoma, what?
Erysipelatooides. It basically looks like an angry, spreading inflammatory rash on the skin.
Okay, got it.
And it is surprisingly common. It affects anywhere from 5–20% of all breast cancer patients.
Wait, really? That is a massive number of people when you consider how prevalent breast cancer is globally.
We're talking about hundreds of thousands of patients.
Right.
And the statistics for this specific complication are incredibly grim.
How bad is it?
Well, historically, skin involvement has been somewhat overlooked in research. It usually takes a backseat to studying metastases in, the liver, lung, or bone.
Sure, the internal organs.
Right. But patients with skin involvement show a profoundly poor response to our standard weapons.
Like chemo and surgery.
Exactly. Surgery, chemotherapy, radiation. Up to 50% of these patients develop disease progression, and the mortality rate exceeds 30% in just the first 5 years following that diagnosis.
Wait, I'm stuck on something here.
What's that?
The skin is our body's biggest physical barrier, right? I mean, it's literally built to be a fortress.
It is, yeah.
It is packed with aggressive immune cells, constantly fighting offcuts, scrapes, bacteria, viruses. So how is a tumor surviving in an environment that is biologically designed to destroy invaders on contact?
See, that paradox is exactly what drove this research. To understand the failure, we first have to understand the normal, healthy immune setup in the skin.
Okay, let's lay the groundwork.
So we have an intricate network of cells, but the absolute most crucial players are dendritic cells, or DCs.
Dendritic cells?
Yeah. They are the essential bridge between the innate immune system.
Which are like the body's general first responders.
Exactly. The bridge between those first responders and the adaptive immune system, which acts like highly trained special ops.
Right. And for you listening, a great way to visualize how these dendritic cells work is a simple acronym, SPP, sense, process, present.
I love that. Yes, SPP. And that first step, sense, relies on these molecular antennas called pattern recognition receptors, or PRRs.
PRRs.
Right. These antennas are constantly sweeping the skin's environment, looking for anything that just doesn't belong.
Just patrolling.
Exactly. And when they bump into a pathogen or a mutated cell, they move to step 2. They process it.
So process it, meaning what exactly?
They essentially swallow the threat whole and digest it. They break it down into these tiny identifiable protein fragments called antigens.
So they chew up the bad guy to get a sample.
Precisely. And then comes step 3, they present.
Present.
Right. They literally travel away from the skin down into the lymph nodes. And they present these antigen fragments to the naive T cells.
So they're basically handing the special ops team a molecular mugshot of the enemy and saying, hey, we found this, go hunt it down.
That is the perfect analogy. And the critical detail here, the thing you really have to remember, is that dendritic cells are the only ones that can effectively wake up those naive T cells to mount a targeted lethal attack.
The only ones.
The only ones. So the core mystery of this paper is right there. If the skin is full of these dendritic cell security guards, why are they ignoring the breast cancer cells actively invading their territory?
Okay, so if I'm mapping this out so far, let's just do a quick recap.
Sure.
Breast cancer spreading to the skin is common and deadly. The skin has guards called dendritic cells that are supposed to sense, process, and present the tumor to our killer T cells. But that system is breaking down.
Completely breaking down.
So let's look at the human data and find out if the guards have just, abandoned their posts entirely.
Well, to answer that, the researchers analyzed tissue samples from 47 human breast cancer patients.
Okay.
23 of them had skin involvement, and 24 did not.
So a perfect comparison group.
Exactly. And they used a technique called immunofluorescence.
Which is using glowing antibodies to paint cells, right?
Basically, yeah. It uses glowing, engineered antibodies to paint different types of cells so you can count them under a laser scanning microscope.
And the results from counting them were surprisingly specific, weren't they?
Very specific. When they checked the general troops, the T cells, B cells, natural killer cells.
The numbers were basically identical, right?
Yeah, identical between the healthy skin and the cancerous skin.
So the immune system had shown up to the battlefield.
It was there. But then they zoomed in on a very specific subset of those dendritic cells. The CD141 positive, HLA-DR positive DCs.
Okay, alphabet soup. We should definitely define those markers for a second.
Good call. So HLA-DR simply confirms it's a myeloid cell, which is just a core part of the immune system.
Right.
But CD141 is the vital marker.
Why is that one so important?
It signifies a specialized type of dendritic cell that is exceptionally good at presenting antigens specifically to CD8 positive T cells.
The cytotoxic T cells.
Exactly. Those are your killer T cells, the ones whose sole job is to destroy tumors.
So what did they find with these specific cells?
Well, in the lesional skin, the skin infiltrated by cancer, these specific CD141 positive dendritic cells were physically present, but their numbers were reduced.
Okay, so fewer guards.
Fewer guards, yes. But more importantly, the ones still standing there were fundamentally broken.
Broken how? Like mechanically?
Well, they lacked activation markers on their surface. Specifically, 2 proteins called CD80 and CD86.
CD80 and CD86.
Right. And without those 2 proteins, a dendritic cell is completely incapable of communicating with a T cell.
Oh, wow. So it's literally like the analogy from the intro. The security guards are physically sitting in the guard shack. They haven't left.
Right.
But their two-way radios, the CD80 and CD86 proteins, have been smashed. And they're asleep on the job.
Yes.
Even if they see the tumor, they can't call for backup.
That is the perfect visualization. But, in science, taking a picture of a smashed radio isn't enough. You have to prove it doesn't work.
You need the functional proof.
Exactly. So they performed functional assays. They literally extracted these live, microscopic dendritic cells from the fresh skin tissues of the patients and brought them into the lab.
And this is where the experimental design gets so clever, to test if the cells could still do step 2, the process step.
Right, from our SPP acronym.
Yeah. They fed these extracted cells what I like to call glowing bait.
Glowing bait, yes.
It's essentially a fluorescent particle. And the idea is, if the cell is functioning normally, it will swallow the bait, digest it, and the cell itself will start to glow under the machine, right?
Under a flow cytometer, yes. And the data was just undeniable.
They didn't glow.
The dendritic cells extracted from the tumor skin couldn't process the bait. They didn't take up the glowing particles anywhere near as effectively as the cells from the healthy skin.
Man. Which means the ripple effect downstream must be catastrophic. If the dendritic cell can't process the antigen, it can't present it.
Right.
It can't wake up the T cells.
Exactly. And the researchers proved that downstream failure, too. They mixed these sleeping dendritic cells with healthy T cells.
Okay. And what normally happens when you mix them?
Well, in a normal, healthy scenario, this interaction causes T cells to rapidly multiply and to produce killer cytokines.
Like biological weapons.
Basically, yeah.
Uh-huh.
Specifically, perforin, granzyme B, and interferon gamma.
I really want to highlight how brutal those cytokines are. Perforin literally punches physical holes in the target cancer cell's membrane.
It perforates it.
Right. And then granzyme B floods through those holes and triggers a self-destruct sequence in the tumor's DNA. It is an incredibly lethal, targeted mechanism.
It really is. But here's the kicker. The T cells that were partnered with the tumor-exposed dendritic cells produced almost none of that ammunition.
Nothing.
Barely anything. And to definitively prove the T cells were now useless, the researchers used two incredible tests: a lactate dehydrogenase, or LDH, release assay, and real-time electrical impedance monitoring.
Okay, I love these assays. Let's break them down visually for the listener because it really drives home how we measure microscopic death.
Sure. Think of the LDH assay like checking for debris after demolition.
Okay, debris.
Yeah, LDH is an enzyme normally kept safely inside a living cell. When a tumor cell is killed by a T cell, its membrane bursts open, and it leaks this LDH into the surrounding liquid.
So by measuring the concentration of LDH in that liquid, you can calculate exactly how many tumor cells the T cells manage to blow up.
Exactly. Higher LDH means more dead tumor cells.
Got it. And the electrical impedance monitoring, that's like cutting a wire on a circuit board, right?
That's a great way to put it. They grow a sheer layer of tumor cells directly on top of tiny microelectrodes.
Okay.
And because the cells are physically covering the electrode, they block electrical current. That gives you high impedance.
Ah, okay.
As the T cells attack and kill the tumor cells, those dead cells detach and float away, which exposes the electrode.
So the electrical current starts flowing again and the impedance drops.
You got it.
But with the T cells primed by our sleeping dendritic cells, what happened?
The current never flowed.
Wow.
The LDH debris didn't spike, and the electrical impedance stayed high. The cancer cells were basically sitting on those electrodes, perfectly safe, totally unharmed by the immune system.
It was a complete systemic failure of the immune cascade.
Complete failure. And it started entirely with the dendritic cell being silenced.
Okay, so let's pause again and synthesize where we are. We're about a third of the way in. We know the body's general immune troops are present in the cancerous skin, but the specific commanders, the CD141 positive dendritic cells, are asleep.
Yes.
Their CD80 and CD86 communication radios are smashed. We've proven in the lab with glowing bait and electrical circuit boards that they can't eat the tumor, and they can't arm the killer T cells with perforin or granzyme B.
Which leaves us with the most important biological question.
Why?
Exactly. Why? The tumors are clearly disabling the dendritic cells, but what is the invisible weapon actually putting the guards to sleep?
Right, and to figure this out, the researchers essentially became microscopic detectives.
They did.
They created what I like to call a tumor soup.
Tumor soup, I like that.
Yeah. They grew breast cancer cells in a liquid medium, let them secrete whatever chemicals they normally produce to survive, and then took that liquid, the conditioned medium, and just dumped it directly onto perfectly healthy dendritic cells.
And those healthy dendritic cells almost immediately lost their activation markers. They stopped working.
Boom.
So the researchers knew for a fact the tumor was actively secreting something into the liquid that caused the blindness.
But, I mean, a tumor secretes thousands of different molecules. So if you have a giant soup of chemicals, how on earth do you isolate the one specific ingredient causing the problem?
Well, they used a brilliant but pretty brutal process of elimination.
Walk me through it.
First, they literally boiled the tumor soup.
Just cranked up the heat?
Yeah. Heat denatures and destroys proteins. So the logic is, if the tumor's weapon is a protein, boiling the liquid will disarm it.
Makes sense.
But when they dumped the boiled soup onto the dendritic cells,
Let me guess.
the cells still went to sleep.
Okay, so we've crossed proteins off the suspect list. Boiling didn't work. What next?
Next, they treated the soup with nucleases.
Nucleuses.
Right. These are enzymes designed to completely shred genetic material, DNA and RNA.
So they're destroying the blueprints?
Exactly. Yeah. Again, the nucleases-treated soup was still highly toxic to the dendritic cell's function.
I love this because it's just so frustratingly resilient. You boil it, you shred its genetics, and it's still functioning.
It is incredibly tough.
Which means the invisible weapon isn't a protein and it isn't DNA. It has to be something tiny, incredibly tough, and heat-resistant.
Precisely. Which brings us to metabolomics.
Oh boy, metabolomics.
It's basically the study of tiny chemical molecules and lipids in our cells.
Okay.
They screened the actual tumor tissue and found a massive, glaring spike in one specific lipid molecule.
And what was it?
Prostaglandin E2, or PGE2.
Okay, let's talk about PGE2, because this is where the spy thriller really takes a turn. PGE2 isn't some exotic foreign poison the cancer invented, is it?
Not at all. And what's fascinating here is that PGE2 is a natural lipid molecule your body makes all the time.
Right.
It is a fundamental part of how your body manages inflammation, fever, and natural wound healing.
So, like, if I cut my finger...
Exactly. When you scrape your knee or cut your finger, PGE2 is there. It helps regulate the immune response so it doesn't get out of control and destroy healthy tissue.
But the tumor is exploiting that. It's overproducing PGE2. And in this specific concentrated tumor microenvironment, the PGE2 acts as a potent immunosuppressant.
Yes.
It physically binds to the dendritic cells and artificially forces them to shut down the expression of those CD80 and CD86 radios. It's basically using our own wound healing mechanism as a cloaking device.
It is a brilliant hack.
Yeah.
And that molecular cloaking device translates directly into devastating clinical outcomes for real people.
Yeah, and here's where it gets really interesting. I think we need to ground this in human reality.
Absolutely.
Discovering this chemical jammer isn't just a cool academic exercise. The paper provides a direct clinical correlation from their patient cohort.
And the data showed a perfect inverse relationship.
Meaning what?
Meaning if a patient had high levels of PGE2 in their skin lesion, they had incredibly low numbers of activated dendritic cells.
The jammer was working.
The jammer was working perfectly. And to make this tangible, the researchers detailed the contrasting fates of two specific patients in the study.
Okay, let's picture what this actually looks like for a patient going into the clinic. Let's look at patient A.
Okay. Patient A's biopsies showed very low levels of activated dendritic cells and very high levels of PGE2 in their skin lesion.
So the immune system was totally suppressed there.
Right. They went through standard surgery to completely remove the visible tumor.
Okay.
But just 3 months later, which is a heartbreakingly short amount of time.
Oh, wow.
Yeah. An MRI and CT scan revealed a local tumor recurrence. The cancer had already come back, right in the exact same spot.
Now compare that to patient B.
Patient B's tissue showed abundant activated dendritic cells and very low levels of PGE2.
No jammer.
No jammer. They also underwent surgery. But at their 4.5-year follow-up, their scans were completely clear.
4.5 years.
Yep, no signs of recurrence at all.
That is a massive difference.
It is. And this wasn't just an anecdote of two people. When the researchers zoomed out and looked at the big picture for survival across the entire cohort, the math was irrefutable.
Let me guess. High DCs equal better survival.
Exactly. Having a high number of activated dendritic cells, which remember requires low PGE2, correlated significantly with better overall survival and dramatically better relapse-free survival.
So synthesizing everything we've learned so far, we've found the guards, we know their radios are smashed, and we finally identified the hammer. It's this lipid PGE2.
Right.
Which just screams one very loud, logical question to me.
What's that?
If PGE2 is the villain putting the immune system to sleep, can we just get rid of it? Like, if we take away the jammer, do the radios turn back on?
That is the million-dollar question.
Yeah.
And it perfectly sets up the final and, frankly, most hopeful phase of the researchers' work.
The therapeutic intervention.
Exactly. But to test this safely and ethically, they had to move out of human samples and into live animal models.
They used mouse models, right? And what I find fascinating is they didn't just look at breast cancer for this part.
Right. They injected mice with either a highly aggressive breast cancer line or a highly aggressive melanoma line.
Melanoma too. Why?
They wanted to see if this PGE2 jamming mechanism was a universal trick used by different types of skin malignancies or if it was just a weird breast cancer quirk.
Smart. So they have these mice with aggressive skin tumors, and the intervention they use next is easily the best part of this entire study for me.
It's so elegant.
Right. They didn't design some billion-dollar experimental targeted chemotherapy that destroys the whole body to kill the tumor. They simply targeted the PGE2 pathway.
First, they used specific laboratory chemicals designed to act as antagonists.
What do you mean by antagonists?
These chemicals physically block the receptors on the dendritic cells, meaning the PGE2 is floating around in the skin, but it can't dock and shut the cell down.
It plays a keyhole.
Exactly.
But then they went a step further, and this is the part I love. They bypassed the experimental lab chemicals entirely and used something incredibly familiar to everyday people.
Celecoxib.
Yes, celecoxib.
For anyone listening, you might know this by its brand name, Celebrex.
Right.
It's a standard, widely available, non-steroidal anti-inflammatory drug, an NSAID.
Right, it's a COX2 inhibitor. Very commonly prescribed for everyday arthritis pain or joint inflammation.
So wait, if celecoxib is just an anti-inflammatory, it shouldn't kill the cancer cells directly, right?
Yeah.
Like, it's not a poison like traditional chemo. It must be doing something to the immune cells instead.
That is the crucial distinction here. Celecoxib works by inhibiting an enzyme called COX2.
Okay, KUX2.
You can think of COX2 as the factory that manufactures PGE2.
Oh.
Yeah. So by giving the mice celecoxib, they shut down the factory. The synthesis of PGE2 completely halted.
And the researchers actually proved it wasn't killing the cancer directly, didn't they?
Yes. They put celecoxib and cancer cells alone in a petri dish together with no immune cells around.
The cancer cells grew just fine.
Wow.
Celecoxib is completely harmless to them in isolation. But inside the living mouse, with its intact immune system, the results were dramatic.
Tell me what happened to the tumors when the factory was shut down.
The skin lesions visibly, physically shrank.
Just from the painkiller.
Just from blocking the pathway. Because with the PGE2 blocked, the immune system woke up. Flow cytometry showed that fully activated dendritic cells flooded back into the tissue.
The blindfolds were off?
The blindfolds were totally off. And right behind them came the effector CD8-positive T cells, now fully armed, expressing incredibly high levels of granzyme B to systematically hunt and destroy the tumor cells.
That is amazing. It worked entirely by empowering the body to do what it was already trying to do.
Exactly. And, to ensure this wasn't just some weird mouse phenomenon, the researchers did one final check.
The bioinformatic data.
Right. If we connect this to the bigger picture, they performed a massive bioinformatic reanalysis.
Meaning they looked at huge databases of human data.
Right. They combed through the Cancer Genome Atlas, which is a huge global database, specifically looking at human melanoma and single-cell sequencing data for basal cell carcinoma.
They were searching for the exact same genetic signatures in completely different skin cancers.
And they found the exact same pattern. Across the board, high expression of COX2 and PGE2 synthesis genes meant bad news and poor survival.
But high genetic signatures for functional activated dendritic cells
meant significantly longer survival. It appears to be a universal mechanism for how tumors survive in the skin.
Man, it is incredible how all these pieces fit together. So for you listening, let's trace this entire journey one last time to really lock it in.
Good idea.
We started with the clinical horror of breast cancer invading the skin barrier. We zoomed in and discovered that our elite security guards, the dendritic cells, were physically there but effectively asleep.
Right, they were missing their CD80 and CD86 radios, which rendered them unable to arm the killer T cells.
Then we followed the researchers' metabolomic detective work. Boiling and shredding the tumor soup to uncover the invisible weapon, a massive overproduction of the lipid PGE2.
That this completely natural molecule was hijacked to act as a chemical jammer. And we saw in human case studies that high PGE2 predicts rapid, devastating relapse.
But finally, we learned that blocking this pathway with existing everyday arthritis drugs like celecoxib could stop the PGE2 production. It could reawaken the dendritic cells, arm the T cells, and visibly shrink the tumors.
It's a profound reminder of how cancer actually operates.
Yeah, it doesn't just divide and conquer in a vacuum. It actively manipulates and hacks our body's own communication network.
But by understanding those hacks, we have real hope. We can repurpose familiar, accessible tools to fight advanced diseases in entirely new ways.
It fundamentally changes how you view the tumor microenvironment.
It really does. And I want to leave the listener with a final thought to mull over, something deeply connected to this, but branching out into evolutionary biology.
Oh, lay it on us.
Well, we discussed how TGE2 is heavily involved in our body's natural, healthy wound healing process.
Right. You cut your finger, PGE2 helps it heal.
Exactly. When you get injured, inflammation and PGE2 are necessary to heal. Now, a tumor has often been described in biology as a wound that never heals. So if tumors are this clever, if they are intentionally hijacking this very normal physiological wound healing process to build an invisibility cloak against our immune system, it makes you wonder.
Wonder what?
What other everyday subtle inflammatory processes in our bodies are nascent, microscopic tumors quietly exploiting to survive right under our noses?
Oh, wow. That is a chilling but absolutely fascinating perspective.
It makes you rethink a lot of basic biology.
It really makes you look at a simple inflammatory response or even an everyday painkiller in a whole new light. The battle is happening right now on a microscopic level inside all of us.
Every single day.
Well, thank you for joining us on this deep dive. Keep questioning the world around you. For you listening, keep looking for the mechanisms behind the mystery, and we'll see you next time.